We've been talking about replication, we've been talking about transcription and translation in the last few videos, and now I want to kind of bring everything back home together and talk about a few minor things when talking about in the chapter regarding molecular genetics. Now, I'm going to explain something, a very important term to you, which is referred to as degeneracy. Now, the molecular information, the central dogma, the DNA that we have, the information that is provided to us by the DNA it's said to be degenerate. And what does that mean exactly? So right here I wrote down that degeneracy of codons is the redundancy of the genetic code. Now, what does that exactly mean? Remember back about the codon chart? Last video we were doing translation, so we were looking at the codon chart, and obviously I don't have the codon chart here anymore, but I'm going to give you some examples. So here's a codon chart that says CGU is equal to arginine, but CGC is also equal to arginine. If you remember from the last video, we saw a lot of redundancies between these amino acids. It's just not that one codon will code for one amino acid. It's in fact that many codons will code for one, or if not many of the many of the codons will code for one amino acid. So in this case, not only is it two codons, but we also have CGA, which codes for arginine, and we also have CGG, which codes for arginine. And this explains the degeneracy of the information that's provided by these codons, CGU, CGC, CGA, CGG. If you can see here, the third codon, the third base pair right here of these codons are essentially, they can be changed. So if you have C instead of U, it's still arginine. So this allows, just in case you have point mutations, right? So let's just say you have point mutations. What are point mutations? Well, these are single substitute-based nucleotides that are changed. So instead of C, I can have A. Instead of A, I can have G. And these all still code for the same amino acid. And this, of course, gives us some flexibility of, if we have any kind of problems reading or translating the final product, which is a protein. And this is pretty cool. But talking about and understanding about degeneracy also leads us to another kind of thing, which is called the Wobble Effect. And the Wobble Effect is also critical to understand. The Wobble Effect explains, or rather shows that, hey, look, we have 64 codons. If we look at our chart, we have 64 codons. And 61 of them code for amino acids. If you remember, 3 of the codons code for stop. I told you that stop does not code for an amino acid. And all the other 61 codons code for a respective amino acid. Remember tRNA molecules, these transfer RNA molecules that take place in the ribosome, they're going to come in and they're going to have the anticodon to the codon, and they're going to put the amino acid into the large subunit of the ribosome. Well, here's something that's very unique. You have a smaller amount of tRNAs in your body compared to the amount of codons. Sorry, 61 codons, not amino acids. There's only 20 amino acids in the body. So, if we have 61 codons that code for amino acids, we have about, perhaps, I think 45 tRNA molecules. And you might say, hey, well, hold on. The anticodon has to be complementary to the codon. The anticodon has to be complementary to the codon, so shouldn't it be the tRNA molecules is equal to the number of codons present? No, actually, we have 45 tRNA molecules and 61 codons, so how is it possible that 45 of these different tRNA molecules can possibly code for 61 codons? This is the Wobble Effect. Here's exactly what happens. In the tRNA molecule, so let me draw my tRNA molecule right here. In the tRNA molecule, so let me just give you an example. Let's use this example right here. CGU. Well, not CG. CG will be the codon. So the anticodon is G, C, and what pairs with U and A. And this tRNA right here that has the anticodon does not have to have GCA to bind on to this CGU. All it has to have is these first two bases. These two bases must match up have the complementary corresponding bases of this codon. So here's my codon, CGU. It's going from 5' to 3'. And this, of course, is going from 3' to 5', my anticodon. And here's my GCA. These two things must match up in order for the tRNA to recognize this arginine and say, okay, time to transfer you to the large subunit of the ribosome. This right here is where it allows some flexibility. So it doesn't have to. It doesn't have to be a U. It could be another base, and the same tRNA molecule will still bind onto this and transfer it onto the large subunit. This is called the Wobble Effect. Let me give you some specific examples, actually. So here's example 1. Uracil, or U, can bond with either A or G. So you can have A or G, doesn't matter. Once again, we see some flexibility here. Here's another example. Inosine can bind with uracil, adenine, and also cytosine, but not guanine. And now you might be asking, what is inosine? Well, inosine is simply a chemically modified version of adenine. So inosine, once again, you should know this. Chances are it won't show up on your AP Bio test, but it doesn't hurt to know. Inosine is a chemically modified base of adenine or adenosine, and it can recognize U or A or C, and you'll still have this one tRNA molecule recognizing either this or this or this, and it'll still bind on and transfer the protein, the amino acid, onto the large subunit of the ribosome. So this is the Wobble effect right here. Now, something very, well, unfortunately, on November 19, 2013, Frederick Sanger passed away. He's a two-time winning Nobel Peace Prize scientist, a genius, really. He died November 19th, which was just a few days ago. He won 2 Nobel Peace Prizes. He won one in 1958 for seeing the amino acids, how amino acid linkage and bondage form the molecule of insulin, and also he's won the Nobel Peace Prize for finding out the discovery of how DNA, how to read the codon, how to read A, T, C, and G. See, we take these for granted, adenine, thymine, cytosine, and guanine. How do we read these molecular bases? Frederick Sanger was one of the founders of genetics, and he showed us how to understand and interpret these weird bases, these purines and pyrimidines that we see. So that wraps it for about the codon, translation, transcription, and replication. Now we're going to talk about some of the things that modify transcription, and this is called acetylation and methylation. So we know that in transcription, we need RNA polymerase to bind on to the promoter region and transcribe. What you may not know is that DNA is wrapped around highly positive charged molecules called histones. So DNA is wrapped around histones, and histones are positive in charge. DNA is negative in charge. So what's going to happen? The histones are going to tightly wrap around the DNA molecule. So let me just draw something here for you. So let's just say, here's my DNA. And wrapped around it is these positively charged proteins called histones. And 8 histones, or an octamer of histones, are referred to something called a nucleosome. You should know that. So DNA is wrapped around histones, and what it does is, if you have highly condensed DNA, the chances of you carrying out transcription is very low. How will the RNA polymerase bind on to the DNA promoter region if it's highly condensed? Well, how do you fix this issue? Acetylation versus methylation. acetyl groups. Add acetyl groups to the histone. If you add acetyl groups to the histone, you're going to make the histone positivity become more neutral, and then the histones can start to come off, and the DNA will be less condensed. So you want to add acetyl groups. So COCH3, you add these groups to the histones, and what starts to happen, you start to uncondense DNA, or I should say uncondense the chromatin, and it allows or it favors for transcription. On the other hand, methylation, you're going to add methyl groups, and this is going to make it more condensed. It's going to make it packed. So you're going to condense, you're going to make this structure highly condensed. And it's usually thought to say that, hey, we're going to have you're not going to have transcription. The chances of you transcribing something are very minimal. So here we have acetylation versus methylation. You should know briefly that acetyl groups, by adding them, you allow for transcription to occur. By methylation, you basically repress transcription. Just keep that in the back of your mind. Nothing so, this is not likely to be a big part of your exam, but this might show up. So please just remember these two. So this is regarding transcription, and now I want to go ahead and go back to replication. We've talked about telomeres, and I've told you that we're going to come to another video and talk about telomeres. So telomeres are found on the chromosome end. So here's my chromosome. We have 23 pairs of chromosomes in the human body on each cell, and in my chromosome, the ends of the chromosomes are referred to as the telomeric regions, or these telomere regions. And these guys basically protect the chromosomes. These guys protect the chromosome after replication. Now you might be saying, hey, what do you mean after replication? What do I have to protect it from, right? I remember replication. Nothing crazy happened. We added DNA ligase, and we've connected the Okazaki fragments together in the lagging strand, and everything seems to be fine. Why are we talking about this? Well, I'm going to tell you about this. I'm going to teach you something right now. Telomeres they're referred to as the end of DNA replication problem. What do I mean by that? Well, as you might remember, when we start off DNA replication, we start off with a replication site or a replication fork. The origin of replication is usually in the center somewhere. We don't start when we replicate, we don't start from here, we don't start from here. We start from the middle. You may remember that from the previous videos. And by doing that, we cause problems with our lagging strand. Let me give you an example. Let me give you an example so you guys can remember. So let's pretend here is my 5' to 3' bottom strand, and here is going to be my lagging strand. Watch. So here is essentially my Okazaki fragment. I've made the complementary bases, and of course here is going to be my primer. Here's my primer. This is going to be my 3' end, 5' end. This should be a brief review for you guys. Remember, primer, we labeled the 5' end. DNA pole 3 will recognize this primer and synthesize from the 5' to 3' direction. But let's say that this is the end of my replication of this huge strand right here. So we have multiple Okazaki fragments right here. We have multiple Okazaki fragments. We have multiple primers. But what happens here? Well, I told you may remember that I said, hey, look, an enzyme comes in, they remove these primers, and you're right, they do remove these primers. They do remove these primers, and they're replaced with DNA, and then we use DNA ligase to connect them together. However, in the end, this last primer over here, in order to take it out and add DNA, if you want to replace it with DNA, you must have DNA before that. But in this case, we don't have DNA here. Obviously, in this section, we do have DNA, so let's just draw it out to make it less confusing. So here's our primer, here's our DNA, here's our primer, here's our DNA. See, we always have this DNA, and it allows us to remove the primers and add DNA and connect them. In this case, this is our end right here. What are we going to do? Well, we've removed this now, so now there's no longer a primer here, so there's no kind of RNA molecules here. However, it's just empty. Nothing happens. We lose this part. We failed to replicate. We failed to replicate the entire segment. So for each replication, you lose about 25 to 200 base pair length. And this is a problem because what's going to happen eventually, after you keep replicating, you're going to start shortening your telomeres. After each cell division, your telomere length will be shortened by this much, and so on, and so on, and so on, and so on, until this cell is considered in a stage of senescence, where this cell can no longer divide anymore. It ultimately has to undergo apoptosis. Apoptosis, programmed cell death, very important terminology. We're going to come back to that when we talk about cancer. But this telomeric shortening causes a huge problem. And, you might say, is this contribute? Well, here's the thing. Here's the fascinating thing, actually. A lot of teachers might tell you that telomeres explain cell aging, because of telomeric shortening, because we keep replicating our DNA, this explains cell aging. It's a good hypothetical reasoning, but we don't have enough scientific data to prove whether or not if telomere or telomeres are a sign of aging or if they are a contributing factor. Of aging. Telomeres. We don't know if they're a sign of aging. If they're a sign of aging, then all this study about telomere and telomerase is primarily not effective in terms of trying to maximize living. I just said telomerase. What is telomerase? Telomerase is an enzyme, actually. Telomerase is very special. Telomerase is an enzyme. Telomerase is an enzyme that will extend the telomeric length. So it's going to extend this by adding sequences of T, T, A, G, G, G to slow down this telomeric shortening. So instead of shortening, you're going to extend. You're going to keep extending this telomeric region, and you're going to allow the cell not to undergo senescence, which is cell aging. This telomerase is only present in adult germ cell line, adult germ cells, cancer cells or tumor cells, but not somatic cells. Somatic cells are body cells. Why is telomerase not present in somatic cells? Why is it present in germ cells? Why is it found in sperm and egg cells? Well, here's the thing. These things are critical. I need to replicate every single portion of my DNA if I'm going to pass it on to my offspring. If I don't have telomerase extending my telomeric region, then the lifespan of this individual or this organism will be drastically shortened. There's been some scientific evidence to prove that. Cancer cells, telomerase and immortal cells have some kind of special relationship. If I have telomerase, for some reason, my cells become immortalized because DNA replication does not affect it, and my cells can keep replicating. This is actually a bad thing. I'll talk about this once again when we talk about cancer. But in somatic cells, we don't have this, so we do experience cell senescence eventually. So after, each cell has a respective amount of cell divisions, but after a respective amount of cell divisions, you're going to see the cell undergoing cell senescence, and there's no telomerase activity. That's not present in somatic cells. Well, you might say, let's just wrap it up, and you might say, hey, well, you know what? Why do we not have it in somatic cells? Well, if we have it in somatic cells, the chances of you having cancer would be higher. At least that's what I believe. Hypothetically speaking, or theoretically speaking, somatic cells having telomerase would be a bad thing, because you would have higher cases of cancer. And actually, there was a scientific study done by a research group where they introduced telomerase into somatic cells, and what they've discovered is that these mice developed autoimmune disorders and such things. So that's it for these things. On the next video, we're going to be talking about regulation of gene expressions in prokaryotic cells.